Welcome to this explainer. Every single time you bite into an apple, you're actually utilizing one of the most incredibly engineered materials in all of nature. Today, we're going to unpack the highly orchestrated, microscopic construction site that was operating inside your jaws long before your teeth ever saw the light of day. We're tracking the unsung hero of this entire story, a highly specialized, incredibly hardworking cell known as the Ameloblast.
All right, before we get into the heavy lifting of how this construction actually happens, let's establish what this process is officially called. It's Amelogenesis. It sounds kind of complex, but it really just refers to the highly organized, step-by-step cellular process of enamel creation. And the primary laborers pulling off this flawless design, those are our specialized cells, the Ameloblasts.
Here is our roadmap for today. We'll start with the roles of the Ameloblast, move into the Secretory stage, look at mineralizing the enamel, explore the maturation stage, and finally wrap up with protecting the crown.
Okay, Section 1. Roles of the Ameloblast, Master Builders.
Looking at the big picture here, these cells hold some massive responsibilities. They're literally multitasking marvels, performing 6 distinct jobs in sequence. First up, they establish the crown pattern during what's called the bell stage, so essentially laying out the architectural blueprint. Second, they act as cellular directors, inducing the adjacent cells to become coronal odontoblasts, which are the cells that build the dentin underneath. Third, they get to work on their signature job, secreting the actual enamel. Fourth, they don't just drop the enamel and leave. They stick around to modulate its maturation. Fifth, they form a protective layer right over that enamel surface. And finally, their sixth role is to actually help form the dentogingival junction once the tooth erupts. So, from the very first blueprint to the final seal against your gums, they are intimately involved every step of the way.
Moving right along to Section 2, the Secretory stage, building the shield.
This is where our microscopic factory officially powers up. During this phase, the Ameloblast cells undergo pretty wild physical transformation. They stretch out, becoming these highly polarized, tall columnar cells. But the absolute game changer happens at their working ends. They develop a highly specialized cone-shaped structure called a Tomes process. Think of it like a biological 3D printer nozzle that actively directs the enamel matrix into distinct rods or prisms. This precise deposition is exactly what gives human enamel its incredibly strong microscopic structure.
Now, as you can imagine, building a complex matrix like this requires a massive amount of raw materials and energy. To pull it off, these cells are absolutely jam-packed with protein-synthesizing machinery. We're talking about a robust Golgi complex, dense granular endoplasmic reticulum, and tons of mitochondria acting as the powerhouses. All of this microscopic machinery works non-stop to pump out the secretory granules that will eventually become your tooth enamel.
Which brings us to a really fascinating quirk about how this stage starts and finishes. The very first and the very last layers of enamel that are secreted actually completely lack those distinct rods, meaning they're prismless. Why does that happen? Well, it's simply because the cell is working without that Tomes process nozzle during those 2 brief windows. The nozzle hasn't fully formed at the very beginning, and it's lost right as the stage wraps up, which leaves a smooth, prismless layer at the dentin boundary and at the very outer surface of the tooth.
All right, Section 3, mineralizing the enamel, hardening the matrix.
Take a look at how vividly this illustrates the transformation. When the initial enamel matrix is just laid down, it's only about 30% mineralized. It's actually relatively soft, full of proteins and water. To fix that, the ameloblasts have to literally pump in massive amounts of hydroxyapatite crystals, while simultaneously using lysosomal vesicles to digest and clear out those soft enamel proteins. It's this intensely active process of swapping out biological scaffolding for pure, hard mineral.
And through this relentless process of stripping away organic material and pulling out water, the enamel hits an astounding final mineral content of 96%. 96! That makes it the hardest, most mineralized substance in your entire body. Honestly, it's an absolute triumph of cellular engineering.
But they're not quite finished yet. Let's look at Section 4, the maturation stage, perfecting the balance.
To get to that perfect 96% hardness, the cells have to execute a highly precise chemical swap. During this phase, we see a massive influx of calcium and phosphates just flooding into the matrix. At the exact same time, there's a selective, highly intentional extraction of the remaining water in specific proteins, primarily one called amelogenin.
And to manage this crazy chemical exchange, the Ameloblasts actually shapeshift. Yeah, they cycle back and forth between two entirely distinct physical forms. Roughly 80% of them take on a ruffle-ended shape, while the remaining 20% shift into a smooth-ended shape.
So what is the point of this shape-shifting? Well, the ruffled-ended cells develop all these microvilli to massively increase their surface area, which is absolutely perfect for pumping huge amounts of calcium ions into the enamel. Then they smooth out, featuring even, straight ends, which alters their permeability so proteins and water can escape. They continuously cycle between these two forms, perfectly fine-tuning the maturation of the enamel based on exactly what the matrix needs, microsecond by microsecond.
Which brings us to Section 5, Protecting the Crown, the final act.
At this point, the active building in maturing is entirely done. But instead of just clocking out and fading away, these cells have one last crucial job. They merge with the surrounding cellular layers to form what is called the reduced enamel epithelium. It's basically a microscopic layer of biological shrink wrap. It completely covers and guards that pristine, newly formed enamel from surrounding fluids right up until the exact moment the tooth finally breaks through the gums.
Because here's the kicker. Once that tooth erupts, those ameloblasts are gone forever. It's a one-time, totally irreversible biological marvel. Your body literally can't make more enamel once the tooth is out. Which leaves us with a pretty wild question to ponder. Knowing the fleeting, incredible life cycle of the ameloblast, from the very first blueprint to that final shrink wrap seal, how often do we take the brilliant cellular engineering behind our own teeth for granted? The next time you flash a smile, remember the microscopic master builders that made it possible. Thanks for joining me on this explainer, and keep exploring the incredible science all around us.